All right, so welcome back. So we're going to talk about filtration now. So we've talked about the nephron a little bit. We've talked about the glomerulus a little bit. So let's get a better look at this. So here on the left-hand side, you've got a picture again of the glomerulus along with the Bowman's capsule. And again, remember the two of those together, both the blood vessel, the glomerulus, and the Bowman's capsule or glomerular capsule put together are called the renal corpuscle. Okay, and it kind of, I like this picture that I found on the internet because it really makes me think of what that glomerulus kind of works like. So imagine this as a glomerulus. It's got lots of holes in it. So again, it's the blood vessel with lots of holes in it. And you've got things in your blood that you want to keep, right? You just want to filter out all the dirt and impurities like on these strawberries. So you have all these little holes that are going to filter all of those things out. Meanwhile, you're keeping, the strawberries in this case behind. Now in the case of your blood, these could be like red blood cells, and you're going to filter out water as well, and along with that water in your bloodstream you're filtering out, you're filtering out, metabolic waste products, things like that. So then it's going to, so imagine if around this colander you have Bowman's capsule, it would maybe be like your sink, if you will, that catches this water along with whatever else is washed out of it, right, is filtered out of it. And then it passes it along down, like, well, in our case, of our sink and our colander analogy, it goes down, the tubes of our plumbing. Kind of very similar here, it's going to go down these tubes of plumbing. The only difference is, unlike in our house where that, that's kind of it just goes into the sewer system. In our bodies, the nephron is also still going to alter the composition of that fluid. So let's take a more of a close-up look of the glomerulus. So what you're looking at here is a little sort of section of it enlarged. So here's the blood vessel, so that's the actual glomerular capillary. And then here are the podocytes wrapping around that capillary. It should remind you of how the Schwann cells created the myelin sheath on neurons, sort of like that, except we're not having like the multiple layers of wrapping. It's just a single, wrap around of the podocytes. And so what you're seeing is that the podocytes will sort of cover up these holes, kind of like if you're kind of thinking of a colander, how you might put your hand on the colander that will help regulate how much water is flowing out of it. That's what the podocytes are also doing. Right? So if we take a look at this image, so here we have our glomerulus. We have the fenestration, so it's a fenestrated capillary, if you remember back to blood vessels. And this will prevent the filtration of blood cells, so those cannot get out. But it will allow a lot of other components of blood plasma to pass through, if this was the only layer. Okay. Now on the second layer, that's the basal lamina of the glomerulus, that prevents large proteins from leaking. So even though they might try to get through here, this will prevent them. Okay, and then you have slits between these sort of little, these finger-like projections. Those are called pedicels of the podocyte. Those slits will prevent medium-sized proteins from filtering out. So the end result between these three different layers, the only things that will get filtered out are like ions, water, and fairly small proteins. So that's why you're not going to find blood in your urine under normal conditions. If you do, it either means that there's damage, so that one of these layers is somehow damaged, so things like red blood cells can leak out, or the pressure is so high that it's somehow squishing the red blood cells out through these holes where they wouldn't normally be able to squeeze through. Okay. So we talk about net filtration pressures. So we have three sort of factors that play into it. And this should remind you of how we had our capillary exchange early in the semester that we talked about. First we have our glomerular blood hydrostatic pressure. And we can really kind of think of that as just in general blood pressure. So this is just the blood pressure moving through the glomerulus, right? That force on the walls of the blood vessel, of the capillary, forcing fluid out. Now we have capsular hydrostatic pressure opposing that. So that's basically the force of fluids that are being filling up that Bowman's capsule, pushing back against the capillary. And then we have blood colloid osmotic pressure. Again, that is going to be the amount of solutes that are in the blood, that are proteins, pulling back on the water, so causing that water to stay within the blood vessel. So as long as glomerular hydrostatic pressure is larger, so as long as your blood pressure is larger than the forces pushing or keeping water in, you're going to have net filtration. Okay. If your blood pressure drops too low, your kidneys shut down because this is not enough to push, the water out and, filter things out, and therefore your blood is not being cleaned, right? All right, so it just shows you that, again, this number has to be larger than those two combined. So the amount of filtrate, and that's what we call it. So when you filter out, whatever you filter out of your glomerulus, that's called filtrate. The amount in all the renal corpuscles of the kidneys per minute is about 125 milliliters a minute. Okay, that's a fairly large amount. Okay, and again, that's an adult male average textbook number. When you think about that, that's just 125 mils a minute. So in about 10 minutes less, you would have filtered a liter of filtrate, right? So like a liter, you have to reabsorb that. Otherwise, you're going to lose way too much fluids. Anyway, homeostasis requires that glomerular filtration rate is constant. So you can see here, if it's too high, you're going to lose things that you want to keep. Okay, meaning you do not have the opportunity to reabsorb them. And that's sort of what this image is. I don't know if you guys remember or have ever watched I Love Lucy, but this is Lucy and this is Ethel, and they are on an old school kind of chocolate conveyor belt assembly line. So chocolates are coming down the assembly line and they're supposed to wrap them in this paper. And what ends up happening is the boss comes out and says, wow, you guys are doing a great job, speed up the belt. So it starts going so fast that they can't keep up with that. So the chocolates are going off the belt, unwrapped, unused, untouched. So now, because they're characters, they're really great characters, they start shoving them in their shirts and their mouths, everything, just to keep them from ending up at the end of the conveyor belt not wrapped. But that's basically what happens. If our glomerular filtration rate is too high, we end up moving things through too quickly and our bodies cannot pick them up fast enough, and then you just lose them in urine. Okay, if our glomerular filtration rate is too low, then basically sufficient waste products may not be removed from the body. Basically, you're not filtering them out in the first place. Oops. What are things that are going to help regulate this glomerular filtration rate? We have 3 different ways. We have what's known as renal autoregulation, neural regulation, and then hormonal. So renal, neural, and then hormonal. Okay, and if we take a look at this image here real fast, again, you've already kind of seen it, but let's take one more look. Here we have our afferent arteriole. Blood is coming in this way. You'll notice there are these cells right here along the perimeter of that afferent arteriole. Those are called juxtaglomerular cells. Blood is entering into the glomerulus. Those are those podocytes covering the glomerulus. So in between those loops, you have another set of cells called mesangial cells. We'll talk about those in a little bit and the role that they play. Anything that gets filtered out is caught by Bowman's capsule, and then it goes down the tubules, first being the proximal convoluted tubule. And then you have here, you can see this, the ascending limb of the loop of Henle, and then the macula densa, these second group of cells here, macula densa cells. These guys will come into play in a little bit. Okay. So let's talk about renal autoregulation first. So it's something called a myogenic mechanism. So basically myo meaning muscle, so it's being generated by muscles, or that's where it's stemming from. So basically what happens is that increased blood pressure stretches the walls of the afferent arterioles. I think that makes sense. We increase our blood pressure, the afferent arteriole where blood is coming in, it gets stretched. In response, the smooth muscles in the walls of the afferent arteriole contracts. So basically the smooth muscles in the wall of the afferent arteriole prevent it from being stretched out too far. But because if this gets stretched out too far, and you have too high a flow into the glomerulus, again, you might end up filtering things too quickly. Too much gets out in there, and you end up with that I Love Lucy kind of scenario. Or worse yet, you may actually even damage the glomerulus. You might actually damage the capillary. Once that's damaged, it's hard to repair. And opposite, if they're not being stretched, they simply relax. So they try to maintain, under normal conditions, a fairly consistent diameter. Right? Tubuloglomerular feedback, so what this is, let's kind of work through this. When glomerular filtration rate is high, fluids move through the tubules very quickly, making absorption difficult. So that's what we talked about, again, the I Love Lucy kind of scenario. We have these macula densa cells in this ascending limb of the loop of Henle, and sometimes you'll see them also in the distal convoluted tubule. They're going to detect an increase in the osmolarity because we filtered. They pick up on those things that our body wasn't able to reabsorb. It's like, whoa, there's a lot of things that are in here that shouldn't be in here. So what ends up happening is those macula densa cells inhibit the release of nitric oxide from the juxtaglomerular apparatus. And the juxtaglomerular apparatus is these juxtaglomerular cells along with the macula densa. They kind of work together to form a single functional unit. Okay, so basically those macula densa cells inhibit the release of this nitric oxide, and nitric oxide causes vasodilation. So again, that vasodilation is the increase in blood vessel width. So if we decrease the amount of nitrous oxide, that would mean that we no longer have vasodilation, or as much vasodilation, which means the blood vessels, particularly the afferent arteriole, is going to contract, which means the flow will reduce, which means less things will get filtered. So we will not be losing so many things down the conveyor belt, down those tubules, and we'll be able to pick up, again, the things that we need back in our bodies. Okay. And then neural regulation of glomerular filtration rate. Sympathetic fibers will cause vasoconstriction of the afferent arterioles. And again, if you think about this, it makes sense. So your fight or flight response, right? Your body is not interested in sending blood to the kidneys to be filtered. If it's a sympathetic nervous response, that means something's attacking you, you got to run away. That means blood needs to be going to muscles, right? Because you got to survive. And afterwards, it'll reestablish blood flow to the kidneys. But at that moment, let's divert that blood. At that point, you're not worried about producing urine. You're worried about transporting oxygen to working muscles. So again, at rest, the blood vessels are maximally dilated, again, because sympathetic activity is minimal. And at that point, that renal autoregulation, where it's those stretch receptors in the afferent arteriole, that is the main regulation of glomerular filtration rate. So like you, not stressed out, this is what's going on. With moderate sympathetic stimulation, because again, there's different levels of stressing out, right? There's, I don't know, forgetting your keys level, stressing out, versus car accident stressing out. Those are very different levels. You might stress out at one versus the other a lot more. Okay, so we have moderate levels of sympathetic stimulation. Both the afferent and efferent arterioles constrict equally. So they both are going to constrict. And basically that simply brings glomerular filtration down equally. Okay, so even that efferent arteriole can constrict. However, with extreme sympathetic stimulation, and I mean extreme exercise, not like, yeah, I got up and changed the channel kind of level of exercise, but like, I don't know, you played soccer that afternoon kind of level of exercise. Or hemorrhage, so we really have that real extreme sympathetic stimulation. You have vasoconstriction of the afferent arterioles to reduce the glomerular filtration rate. Okay, again, kind of what we kind of talked about it there. So it simply is going to lower the urine output and permit blood to go to the tissues that need it. Right? And then we have hormonal regulation of glomerular filtration. So that's atrial natriuretic peptide, so ANP, that'll help increase our glomerular filtration rate. So the way this works, and maybe we can tease this name apart. So atrial, referring back to the atria, so stretching of the atria that occurs with an increase in blood volume causes the hormone to release. Okay. One of the things this hormone does, in the case of glomerular filtration rate, it relaxes those glomerular mesangial cells and it increases capillary surface area, increasing glomerular filtration rate. So let's take a look at that in this image. So that ANP, the stretching of the atria, recognizes that there's increased blood volume, causes ANP to be released. Remember, we had these little mesangial cells, those little cells in the loops of the glomerulus. So basically, those are going to simply relax, and which means that those slits right here are not going to be as stretched. So kind of think of like, I don't know, like if like a rubber band kind of thing, like if it's stretched, like the little slits between the two sides of the rubber band are really narrow. Obviously, it's harder to have something fit through there versus when rubber bands relax, it's wider. So kind of like that. So those mesangial cells relax, the slits open up, that increases the surface area and helps increase glomerular filtration rate. We have something called angiotensin II, and we'll talk much more about this when we get to the pH and ions portion. It reduces glomerular filtration rate, so it does the opposite. And angiotensin II, it's a potent vasoconstrictor, so it narrows both the afferent and efferent arterioles, reducing the glomerular filtration rate. All right. So hopefully that was fairly clear to you, but if you happen to have any questions, please do not hesitate to ask. I'll see you next time.